Defective T-cell signaling, cytokine production, or cytokine action can cause immunodeficiency. Virtually all adaptive immune responses require the activation of antigen-specific T lymphocytes and their differentiation into cells producing cytokines that act on specific cytokine receptors. Several gene defects have been described that interfere with these processes. Thus, patients who lack CD3 gamma chains have low levels of surface T-cell receptors and defective T-cell responses. Patients making low levels of mutant CD3 epsilon chains are also deficient in T-cell activation. Patients who make up a defective form of the cytosolic protein tyrosine kinase, ZAP70, which transmits signals from the T-cell receptor, have recently been described. Their CD4 T-cells emerge from the thymus in normal numbers, whereas CD8 T-cells are absent. However, the CD4 T-cells that mature fail to respond to stimuli that normally activate via the T-cell receptor, and the patients are thus very immunodeficient. Another group of patients show a lack of interleukin-2 production upon receptor ligation, and these patients have a severe immunodeficiency. However, T-cell development is normal in these individuals, as it is in mice, which mutations have been made in their interleukin-2 genes by gene knockout. These interleukin-2 negative patients have heterogeneous defects. Some of them fail to activate the transcription factor NFAT, which induces the transcription of several cytokine genes in addition to the interleukin-2 gene. This might explain why their immunodeficiency is more profound than that of mice whose interleukin-2 gene has been disrupted. Interleukin-2 deficient mice can mount adaptive immune responses through an interleukin-2 independent pathway, possibly involving the cytokine interleukin-15, which shares many activities with interleukin-2. Nevertheless, they are susceptible to a variety of infectious agents. And that's defective T-cell signaling, cytokine production, or cytokine action can cause immunodeficiency. Have a great day.